we will switch gear a little bit because we have been interested in epilepsy and treatment for epilepsy in an orphan disorder called tuberculosis, TSC. Switch slide. Is that working? Actually, I'm going to move it here. Last 10 seconds. So anyway, the next slide will show you why TSC, what is important about TSC. So this is an orphan disorder, and the patients have genetic mutations, and the most dramatic phenotypes of these diseases is actually epilepsy. 90% of them have severe seizures. And it might not work. It works. Thank you. Yeah, I'll do reason to. So 90% of them have seizures. They're diagnosed around a medium of 3 months of age. They will have seizures for life. And we actually know the cause of seizures. This mutation occurs during fetal development in brain cells and lead to this kind of brain malformations. One of the standard of care in 10 to 15% of the patient is to remove these brain malformations. The other standard of care is the drug sirolimus, but both of them don't work very well and have terrible side effects. So we really need better treatment for this patient. On top of it, there are a lot of comorbidities with seizures that includes insomnia, learning disability, and really behavioral problems like anxiety. So there is really a cry by the patients for better treatment. This is, although it's an orphan disease, there are about 30 to 40,000 people who have anti-epileptic drug-resistant epilepsy in the U.S. The cost is actually quite enormous, $16,000 per patient per month, which gives you a market opportunity of $4 to $5 million a year. So I've been working on TSC for the past 12 years, and I have very close ties with the TS Alliance. I'm on the advisory board for the preclinical consortium and look at their leads, clinical leads. And we can have a direct access to quite of the patients. The TS Alliance oversees 68 TSC clinics across the U.S. And then competitions, well, we're not the only one to realize that we will find a treatment in 5 to 10 years for TSC epilepsy. And there was a new kid on the block, Neurolexa, that was approved in 2020. That drug does not work well. 20% seizure reduction, terrible side effect. We may have another drug, the MEG blocker, but as you know, that drug has also terrible side effect and actually came from our own work. So that's why it's just complicated. And I'm sorry that I don't have the animation. I forgot to put it in. But basically, if you focus on this thing, they have mutation that leads to increase in mTOR activity. It's a really important intracellular hub. That's the target of the standard of care, everolimus. What mTOR does, he makes big cells. That leads to seizure. So really, what I want to show you in a minute is why do they get seizure? What does that mean? So here's a normal cell. It's a little small here. This is a neuron in in the mouse cortex. We have them. They're beautiful. They are processes. They receive and they make connections. It's a normal one. In the disease condition, that's the one. Here it is. Very clear. The cell is bigger. mTOR does that. Because there's an increase in protein synthesis, they acquire the wrong channels and they're hyperexcitable. One of these channels is HCN4, which makes them pacemaker like your heart. The other thing that is obvious is connectivity. We're all making connectivity in our network today, but in this case, this is not good. It's over-connected, and it goes all over these cells, send projection to all over the brain, and trigger or make the seizure travel farther. So what we did is that we blocked these increase in protein synthesis by overexpressing that little molecule 4-ABP. We turned that cell back into that one, and that reduced seizure activity. And this is a target for gene therapy. Local injection with neurosurgeons, since we have local brain malformation. The other things that we found accidentally is the overexpression of that molecule, filamin A. So filamin A is actually acting like a VC. It has a lot of binding partners inside the cells, and it allows the wrong partners to be expressed in that cells, which triggers excitability. When we blocked filamin A function or knock it down, we turn it back to that, and that blocks seizures. So that's what we did. And the work in mice is translatable to human because the same pathology, and we monitor seizure activity. And we're at the stage of seed where we need really money to make new drugs that are better and that we will own, and having a composition of matter patent. And we would actually also like to have, I guess I lost my slide, the last slides. And we also would like to eventually try gene therapy, which takes more time. Thank you for your attention.